Welcome to the first in a series of outreach events that the Technology Development Coordinating Center is presenting in May to highlight the work of a number of NHGRI funded grantees in the Genome Technology Development Program. I'm going to give a quick intro to the TDCC, and then we'll get started with the program. The TDCC was founded about 2.5 years ago to bring together programs from across NHGRI that are related to genome technology development, including new sequencing technologies, synthesis technologies, a wide variety of genomic approaches related to characterizing the genome at large scale. And our purpose is to help spread the word about those new technologies, as well as provide some coordination, pilot funding, and discussion formats for the grantees. This is, you can get more information from our website, genomeTDCC.org. This is the first of 4 sessions that are planned this month. The next one is on single molecule protein sequencing on Friday. And then next week, we have 2 sessions on single cell methods and genome-wide regulatory analysis. I know many of you will have signed up for all 4 of these. Welcome. Look forward to seeing you back. If you happened to cross this one alone and would like to see information about those, please check out the genomeTDCC.org website. For today, this is a webinar format. That means you can enter your questions in the Q&A box on Zoom. We will be having each of the 3 speakers present their talks, and then we'll use time at the end of the session for Q&A. We invite you to go ahead and enter your questions as they come up, and we'll collate them and present them when we get to the end. Today, we have 3 terrific speakers who are going to be talking about a really exciting and emerging area of biology, which is RNA modifications. This is a topic that hasn't gotten as much attention in the genome world because these modifications are difficult to study, and yet they have really important contributions to our biology and how the regulatory program of the genome is interpreted. So our first speaker today is going to be Dr. Ya-Ming, who is professor of biochemistry and molecular biology at Thomas Jefferson University. She's been there 30 years, which is quite an accomplishment in longevity in the academic world. She works on the fundamental problem of how RNA is translated into protein, which sounds like a simple matter of delivering the next tRNA, delivering the next amino acid to the ribosome. But as you'll hear, is much more complicated than that. And the nuance and sophistication of this process is amazing. And she's made some great contributions in this field. Ya-Ming, welcome, and lead us off, please.
Thank you. So I'm really happy and honored to be the first person to start this session. I think the session is going to be very exciting. It's going to lead you to the frontier of the state-of-the-art information about what RNA modifications are, what do they do, and how they impact human health and disease. So basically, we're talking about the RNA world. The RNA world is expressed, is expressed not just only by nucleotide sequence, but also by the diverse set of post-transcriptional modifications that are installed in individual nucleotides. And each modification can have an impact, specific function on the messenger RNA or on the tRNA or on the ribosomal RNA. So we'll go into detail. So RNA modifications are already playing a very important role in our human health. During the pandemic of COVID-19, many of us have been vaccinated with this messenger RNA vaccines. And in this messenger RNA vaccine concept, we actually have many RNA modifications. So starting out with a normal uridine, and there can be modified into pseudouridine, and then the pseudouridine can be further modified with M1 at the N1 position of this pseudouridine with a methyl group. So it's M1 pseudouridine. So during a chemical synthesis of this spike protein of the virus, SARS-CoV-2, a gene will be synthetically made, and where in each U will be replaced with the M1 pseudouridine modification. And when the synthetic gene is packaged, delivered into human cells, the messenger RNA is released, and this will be the substrate for protein synthesis on the ribosome. So the ribosome will generate lots of spike proteins of the virus, and it will activate the immune system to generate antibodies that target the spike protein. And when viruses are infecting human cells, it will be the spike protein that will be the first protein to anchor onto the human cell, and antibodies at that time will recognize the spike protein on the virus and will destroy the virus. So that provides the fundamental defense mechanism against the virus. So the RNA modification is already playing an important role in our human health, and where you and I are both healthy today. In fact, RNA modifications are found in all three major types of RNA. It is in the non-coding RNA, such as tRNA. In tRNA, there's an L-shaped structure with an amino acid attached to one end, anticodon to the other end. And when the tRNA reads the correct codon, the amino acid will be delivered to that codon. And this whole process, the accuracy, the speed, the quality depends on RNA modifications, which I will go into later. In ribosomal RNA, these are the RNA molecules that support either the decoding center or the peptide bond transfer activity active site. And both RNAs are large RNAs, and they are peppered with lots of modifications. Even in eukaryotic messenger RNAs, where we start out with a modified guanosine, that is M7, methyl group attached to the 7th position of the guanosine, starting out with the cap structure, followed by 5' UTR coding sequence, 3' UTR, and the poly A tail. Again, throughout the entire sequence of the messenger RNA, there are modifications that regulate the fate and function and activity of the messenger RNA. So the important message I would like to convey is that all types of modifications converge. They talk to each other at the site of ribosome protein synthesis. So tRNA modifications encountering a codon containing a modification in the messenger RNA, supported by translation that is regulated by modifications in the ribosomal RNA. They all perform crosstalk to each other at the site of each decoding event. So this crosstalk is important, that regulates the quality, the speed of protein synthesis. And indeed, the quality and the speed of protein synthesis determines the quality of life, the fitness of cell fate during each stage of the life stage. So in the presence of modifications, usually slows down the decoding event. And when you perform a slower translation, ribosome is more accurate. Each amino acid is precisely matched with the codon. In contrast, when the ribosome performing translation without modifications, it usually goes like a hasty speed. And the accuracy is reduced, and the ribosomes can sometimes misincorporate wrong amino acids that don't belong to the right codon. So resulting in wrong amino acids, misincorporated proteins, and that usually confers shorter lifespan. So quality of protein synthesis is important, and that quality is determined by the crosstalk of all three types of RNAs that contain modifications. So this is to show you that the modification chemical diversity is huge. Like in tRNA, which we usually have 10 to 20% of the molecule modified, there are many different types of modifications, starting with a simple one like a methyl group or acetylation to the more complicated structures such as MS2, T6A, that sort of QOC modifications. So these are very complex structures that are important for determining the quality of tRNA translation. In ribosomal RNA, many modifications are concentrated in the decoding center. Again, they are They have a diverse structures, even in messenger RNAs. Although the stoichiometry of modifications in messenger RNAs is usually not as high as those in tRNAs or ribosomal RNA, they nonetheless are specific and they define the function of the messenger RNA. And so far, we are very comfortable to know that these are the modifications that are present in messenger RNA. These are usually simpler chemical structure, usually methylation or pseudouridination. So this is to give you an idea of what modifications can do and how they respond to stress. For example, there is a conserved and static modification that's been conserved throughout the evolution of life. And this is concerning a guanosine with the M1 methylation to the N1 position of the guanosine nucleotide on the 3' side of the anticodon. Even though it's not directly involved in anticodon-codon pairing, it determines the quality of decoding. And we show in my lab that this methylation is important to make sure that there's no frame shifting. If there's a frame shifting, that results in premature termination of protein synthesis and leads to cell death. Another role is that this methylation determines the efficiency of charging. If there's methylation removed, the charging is deficient, tRNA stalls and ribosome stalls, and nothing goes on. So eventually leads to cell death. And the more complexity is that this methylation is conserved in 3 families of amino acid, proline, leucine, and arginine. So these are conserved patterns of modification that makes decoding even more complex and has even additional layers of regulation. Another modification that is conserved and static is occurring in the ribosomal small subunit. There's a ribosomal RNA that contains 2 consecutive adenosine residues, and in each, it undergoes 2 rounds of modification. The first round occurring at the N6 position with 1 methyl group, and the second round with the same site of N6, but there's a second methylation. So these modifications have occurred throughout the evolutionary domain. It's always there, and particularly it occurs in the small subunit near the P site, which is regulating the initiation event of protein synthesis that regulates the downstream process of protein synthesis. It is important for initiation of protein synthesis. There are modifications that are conserved but reversible, meaning that they can be put on and can be taken off. An example is in eukaryotes, particularly methionine tRNA. There is an adenosine at position 58 in the corner of the tRNA L shape that is modified by this complex. And in low glucose, there would be an eraser, a protein that removes this modification. So the tRNA now becomes empty at this position. And this will impact how this methionine tRNA performs initiation, how this methionine tRNA can also perform elongation. So there are 2 types of methionine tRNAs. One responsible for initiation that will be affected, another one responsible for elongation that will be also affected. There are also modifications that will bring out different outcome, even though it's the same chemistry. If there is an A adenosine following the cap structure, that A is typically modified with a M6A, but that only occurs when the ribose of that A adenosine is already methylated with the 2' the sugar position. When that is this M6A will be performed by one methyltransferase that is distinct from the other methyltransferase that will put in M6A. This will occur downstream during the coding sequence and in the coding sequence and near the stops. So the outcome is different. In the first case, when methylation occurs near the start, it will make the messenger RNA more stable. In contrast, when the methylation occurs during the coding sequence or the stop, the messenger RNA is less stable. And when this methylation is removed by an eraser, it will confer the loss of control of cells for body weight, and the organisms tend to become obese. But in the other case, when this methylation is removed by another eraser, it will lose the control of embryonic development, and the organisms will lose the control of fertility. So the outcomes are quite different. There are also modifications that respond to stress. For example, in bacteria, when it encounters a hypoxia condition, low oxygen, it will activate a pathway that converts uridine in two steps of reaction to carboxy methoxy 5 position of uridine. This modification is CMO5, specifically occurring at the wobble position of the anticodon. And this modification enables the wobble base pairing better and more efficiently translated. When a hypoxia condition starts, bacteria activates this modification. It will selectively translate messenger RNAs that are enriched with codons that will be better translated by this wobble modified anticodon. So it allows bacteria to selectively translate genes that will support growth in hypoxia. So bacteria survive better. So this is an example. Another example is that there are also cases where one modification regulates the activity of the second modification. This is in the case of M1G. When M1G is present, it allows this modification, which we just talked about, CMO5, to read all 4 nucleotides. In the absence of this methylation, it will not be able to support this reading of the wobble through wobble with the pyrimidine ending wobble positions. So these codons will not be read by this tRNA, and cells will require another dedicated tRNA to read these codons. So that creates intricacy of how translation can occur during codon sequence bias. Also, during disease states, throughout many post-transcriptional transcriptome analysis, we now know that modifications or RNA modifications will undergo profound and genome-wide landscape changes. So for example, for modifications involving M1A, M5C, M6A, we know the writers. We also know the erasers. And each time there is a disease state that undergoes stress, that writers respond to that stress, erasers respond to that stress, and it will undergo a programmatic change of the landscape of modifications. So writers will put in new modifications, erasers will take out some existing modifications. You end up with a different profile. In another group, like pseudouridine M7G and inosine, we know the writers, but we have yet to identify the erasers. So that remains a challenge. But nonetheless, there would be that we have found changes, programmatic changes of these modifications. And these are particularly strongly associated with cancer, with neurodegeneration, and metabolic disorders. So this highlights the importance of how modifications can impact disease. So one modification in terms of enzymology can be installed by 2 different enzymes. In the case of M1G, we found that in bacteria, it's installed by TRIM-D, in eukaryotes by TRIM-5, and they have distinct structures. They are not homology. They are not homologous in structure or in sequence. For TRIM-D, the mechanism is binding this methyl donor in a bent shape. This is adenosine, this is adenosine, this is methionine. They are bent. The 2 moieties are bent to each other. In TRIM5, this methyl donor is open in a straight conformation. TRIM-D recognizes only part of the L shape. TRIM5 recognizes the entire L shape. So the mechanism of tRNA recognition is distinct, and the response to stress is distinct. There are also examples of one modification, that's pseudouridine modification in messenger RNA, that is installed by 3 different enzymes. In human, in the eukaryotes, higher eukaryotes, is PUS1 correlated to a tRNA pseudouridine synthase enzyme TRU-A. There's a PUS4 correlated to TRU-B in tRNA pseudouridination, and PUS7, which has a much bigger structure. So they all share the same pseudouridine synthase domain, but they have different structures for RNA recognition. For PUS1, the modification for pseudouridine is installed at the bottom of a stem loop structure. For PUS4, it is in the loop structure. For PUS7, it prefers a linear and unstructured sequence. So they are all different, and their response to stress is different. So in summary, what I like to say is remember that all types of modifications converge. They talk to each other during the important step of protein synthesis on the ribosome, and that determines the quality of life. All right, but today we know that modifications is important, critical to human health, and because mutations and modification enzymes are frequently underlying the various diseases that we know, and the major challenge that remain and that you will hear in the next two talks is about the methods and the framework of how to elucidate the modification code. So I will end here. I thank people in my lab. I thank my founding structures and organisms, and also I thank my collaborators for supporting the work that we are moving forward to. Thank you.
Wonderful. Thank you so much, Ya-Ming. And I notice you have Sara as one of your collaborators there. Sara Rouhanifard is our next speaker. She is assistant professor of bioengineering at Northeastern University. She's been there since 2019. Before that, received her Ph.D. from Albert Einstein College of Medicine. And she's going to introduce some of the methods that are used, as Ya-Ming said, to decode and understand these and determine these modifications. Sara, take it away.
Hi, thank you to Mark and the TDCC for organizing this event and for the invite. And thanks to Ya-Ming for a really amazing introduction to the important biology of RNA modifications. So now I'll start to tell you about some of the challenges and opportunities, I think, in mapping RNA modifications, specifically in messenger RNAs. This is what the focus of my talk will be about. So just really briefly, my lab is interested in understanding how RNA modifications drive gene expression. So we do this by mapping and identifying RNA modifications de novo, and we use a technique called direct RNA sequencing by Oxford Nanopore, which I'll talk about later in this talk. And then we like to quantify the occupancy in silico, and then we like to detect them inside of cells in order to understand the physiological function of the mRNA modifications. We're also really generally interested in building RNA imaging tools, which I won't talk about today. So Ya-Ming mentioned this before, but we have over 150 different RNA modifications that have been found throughout all classes of RNA. The majority of them have been discovered on our ribosomal RNA as well as our tRNA, because these are, one, really heavily modified, and two, because the majority of our total RNA inside of cells comes from tRNA and ribosomal RNA. So you have a lot more material to work with when you're doing analytical techniques. So it's a lot more amenable to identification and mapping. But mRNAs represent only 3 to 7% of total RNA by mass. So just this little green area here and this little green area here. But mRNAs are a really interesting target for RNA modification because they have the potential to fine-tune our protein expression and respond really quickly to environmental factors. And that's why what I'll talk to you about today is there's challenges with finding them in mRNA modifications, but perhaps it's worth it because we want to find something really cool that is going to affect our protein expression. So my short talk today is going to be about general strategies for mapping mRNA modifications, and I'll give you some specific examples in the end about how our lab is approaching it. But these are very big picture, broad things that you would think about if you were pursuing a project that you wanted to map mRNA modifications. So I tried to break down what the requirements might be if one wanted to pursue this challenge. So the first thing you want to know is which modifications are even there. I mentioned on a previous slide that there are 150 possible RNA modifications that we know of. We're not even sure which one of them are found on mRNA. I'll show you on the next slide the ones that we know already are on mRNA, but imagine coming up with a technique where you're not sure what the chemical makeup is of all of your mRNA. So that's the type of challenge that we're looking at. So you're going in completely blind. So the first thing you need to do is the modification there at all, so I know what to hunt for. And this is going to be done typically by mass spectrometry, which you'll hear about in the next seminar. So the next thing you want to do is figure out which technology you want to want to use. These certainly aren't the only ones you could use, but I'll break my talk down into Illumina short read sequencing, which converts your RNA into cDNA before the final readout. And then the Oxford Nanopore platform, which is taking your RNA directly and analyzing it directly on the native strand. But this also has some challenges because you need to figure out how to interpret the signal that comes out of that platform. And then finally, I'll end with the types of controls that one needs to consider if you're thinking about mapping the epitranscriptome. So which modifications are there? That was the first requirement. I've added the structures of 6 different RNA modifications that have been identified on human mRNA, and new ones are coming out every day. New discoveries about which RNA modifications might be there. You'll find out about them, like there's a few more added every year as the technology is developing. So it's kind of the Wild West out there. So the number below these structures, so for Psi, I've written 0.29% to 1.04%. That's just an example of the percent of the canonical nucleotide that would be modified. So out of 100% U, only up to 1% of those U's will be converted to Psi. And likewise for all of these. But the point is not really that it's a low abundance. It's just that even though they're low, it's happening right on your mRNA that's going to get translated to protein. So the potential is there that it could really modulate your protein expression. So the next thing we want to consider, first we want to know which ones are there. So far I've presented 6 or 7 of them that we know might be on the mRNA to begin with. Next thing you want to do is think about what technologies you might want to use. So I've highlighted here inosine and M6A, and these are the first 2 RNA modifications on mRNAs that we've known the most about, and there's a special reason for that. So for inosine, you can see this image on the right here. That's an A converted to an inosine, so an adenosine converted to an inosine. Now, A base pairs with T, but inosine is a guanosine analog, so it's going to base pair with cytidine. So when you have a Watson-Crick base pairing change, that makes it amenable to a sequencing technique without chemical intervention. So that's the reason why inosine, we know a lot about inosine for now. M6A is also a unique one because there was an antibody developed that could probe M6A directly. So once you have a probe that can target your RNA modification, you have a lot more techniques available to you that you can do something with. So inosine and M6A are sort of unique in that there's an antibody and they change Watson-Crick. Not the case with all of the others, all of the 150 others. So for the other ones, the techniques that we typically go after are 1, chemically assisted Illumina sequencing, which I'll talk about in the next slide, and also the direct RNA sequencing by Oxford Nanopore. So for chemically assisted Illumina sequencing, you start off with your mRNA strand at the top here, and the N with the red circle around it is your modified nucleotide. So you're going to introduce a chemical that binds specifically to that nucleotide that you're interested in. Now, you could do a biotin enrichment. I won't talk about that right now, but the general idea is when you have this bulky chemical group there, if you do reverse transcription, which is required, you have to create a complementary DNA in order to do Illumina sequencing. When you do reverse transcription, your reverse transcriptase is going to read through your RNA, and then it's going to stop once it reaches that bulky chemical group that's attached to your nucleotide that's modified. Alternatively, it can lead to a deletion at that nucleotide and then keep reading through, or it can lead to a misincorporation. So the wrong nucleotide that's complementary to your modified nucleotide. And this is the result of that chemical bulky group being there. So by sequencing, this could lead to, you see nucleotide, 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 and then a stop. So it ends there. So that's how you know the modification is there. Or you have something that's skipped, which is a deletion, or a miscalled base. So these are the 3 different options that you would have for a chemically assisted Illumina sequencing. So now if we switch to the direct RNA sequencing, in this case, this is the Oxford Nanopore platform. And what we're looking at is a strand of DNA that's being passed through a biological pore. So as it passes through, the instrument is going to measure an ionic current disruption that is associated with individual bases. And that's what you're seeing in this image right here. In real time, it's going to read the ionic current disruption and associate that with a base. So A, G, C, and U. Now, if we take this, what we call a squiggle, and overlay it with a strand that's modified. So let's say the red signal here is an unmodified strand, and the blue is showing a modified strand. You can see that in the center position, you have some of the ionic current that's disrupted. So it's not coming up in the spot that you expect it to. And that can be indicative of an RNA modification or some kind of deviation. So when we analyze Oxford nanopore signals for RNA modifications, there's two ways that we do this. One of them is by base miscall analysis. So as it's passing through, it could miscall your RNA modification as another nucleotide, or you could simply interpret from the current signal directly. So these are the two major ways that you could use direct RNA sequencing to interpret RNA modifications. So the next thing is thinking about what controls one would use for doing either of these techniques. But I'll focus mostly on the Oxford Nanopore platform from now on. So controls are complicated for mRNA modification. So first, you would think about your negative control, which could be an enzyme knockdown. So the problem with this is that the enzymes that modify mRNAs also modify tRNA and ribosomal RNA. Also, we don't know if there's redundancy in those enzymes. So if you knock one down, one other one might recover. The other thing that you could do is something called an unmodified transcriptome, which I'll talk about. This is a convenient option where mRNA is converted to cDNA and then back to a clean copy of RNA. And this is a very convenient negative control. For your positive controls, you could use a known sequence that's validated or verified by mass spec. So often ribosomal RNAs can be used for this, but this doesn't work for the Oxford Nanopore platform as a control for mRNAs. Alternatively, you could use a known sequence that's generated by solid phase synthesis. So using a synthesizer and then planting a modification in a specific site. The problem with this is that it's super expensive and the quality control is complicated. Doing RNA synthesis is not as easy as we think about for DNA synthesis. You could also do a known sequence that's generated by in vitro transcription. So in this case, you would replace, if you're studying a uridine modification, you can replace every single U in a previously generated sequence with a modified U, but that would preclude the analysis of sequences that have greater than one U. So I'm just going to give you some examples of how we use the Oxford Nanopore control in our methods. So I mentioned we have a unmodified control that we use. So if you take your native mRNA and you do a library prep with that, you pass it through the nanopore and you get your signals. You could take that same native mRNA, reverse transcribe it to make cDNA, and then in vitro transcribe it to make a clean copy. So the IVT is done with canonical nucleotides. So what this looks like with base calling errors is your IVT RNA is going to be clean. You have some sporadic errors that occur, and this is just standard error, but then you have a systematic U to C base calling error at the site of a modification. And that's true of all three of these genes that have a modification in the center position. So the IVT is a clean copy, and then the blue is indicating a U to C mismatch error. So another thing that we do is we generate synthetic controls. So these are generated by purchasing a 15-mer RNA that has a pseudouridine planted in the center of it. And this is done in collaboration with Ya-Ming, who spoke previously. So we'll use a splint ligation to generate a very long RNA oligo that has a pseudouridine planted in the center of it. So we wanted to see how quantitative this base calling error is. If it was 100% quantitative, you would expect these bars to be blue, completely blue, but we see it being called as C as well as U. And for each of these genes, the percentage that it's getting miscalled as a C is different. So we know that the method is not quantitative as is, but what we can generally say is that it's always undercalling the psi. So we can make some semi-quantitative measurements from this data directly without doing any kind of intervention. We can say that everything that has a U to C error of higher than 40% is very highly modified, or it has high occupancy at that site. And that could lead us to some biological insights already. But what we can do is we can take those controls and extract additional features to correct for that error using machine learning algorithms. And when we do that, we can achieve greater than 90% accuracy in quantifying the sites. So we applied the machine learning algorithm to direct RNA sequencing from 7 different human cell lines from different parts of the body. And we found that the occupancy for a given site changes across cell lines, suggesting that there's another level of regulation happening there. So I'm going to take a quick shift, just super quick, because my lab is very interested in learning the biology of RNA modifications. So once we have this data, and we can see which sites are very highly modified, our interest was applying an imaging technique to see what they're doing inside of cells. So a technique that my lab uses a lot is called single molecule RNA FISH. So if you have your target mRNA molecule, really briefly, you design short 20-mer DNA oligonucleotide probes that have a fluorophore on the end of them. And you'll design like 20 or 30 of these tiled on your mRNA. So individually, these would produce stickiness and bright spots all over the place, but collectively, they'll form a single crisp spot on your cell. And each of these dots is a single RNA molecule. So what we did was we designed FISH probes against these sites that were almost 100% modified so that we can learn some biology by the very high occupancy sites. So we're treating every single site as a modified site. So this is just some preliminary data from my lab that I thought would be interesting for the talk. So what we did, we took wild type HeLa cells and we probed them with FISH probes for some of those sites. So AK2, just for an example, if I go back, AK2 is right here. So it's showing up as 90% U to C mismatch error, which means it's almost 100%. Every single site on there is going to have a pseudouridine on it. And then we generated a knockdown cell line for the enzyme that creates that modification. And we tested this for 3 of the genes that were showing up in our nanopore data. And some of the interesting patterns that we've seen so far are, the most obvious one is here. So we'll look at nucleus versus cytoplasm. And for these 2, you see there's no difference. But at the bottom, you can see that for GT3, these cells are burstier, so more transcriptionally active. That's the site that the polymerase has sat down on the DNA, and it's firing off a whole bunch of RNA molecules. So using this technique and the data that we've collected from Oxford Nanopore, the semi-quantitative data so far, we can start to ask some questions about how the RNA is behaving inside of the cells. And with that, I'd like to thank my lab and my collaborators, and thanks to TDCC for organizing the event.
Thank you so much, Sara. Our last speaker is going to be Sigrid Nachtergaele from Yale University. She's an assistant professor in molecular, cellular, and developmental biology there. Sigrid got her Ph.D. from Stanford, and her lab is focused on understanding how RNA modifications influence cell signaling and function. So Sigrid, take it away.
Thank you. Thank you for inviting me to participate in this great panel. I'm really honored to sort of follow up on these fantastic first 2 speakers. And really what I'm going to try to do here is sort of build on what both of them have talked about and talk a little bit more about how we quantify RNA modifications and measure their stoichiometry. And I'll tell you a little bit more about what I mean by that, but just a quick disclosure, I am on the scientific advisory board of an RNA-related company, so I'm just listing that disclosure here. So the lab here at Yale, we've been here about like 3 years now, and we're really interested in a lot of different aspects of RNA modification biology. And so that's sort of schematized on the slide here, but essentially we're interested in everything from discovering new RNA modifications to, their mechanisms and understanding how they impact human health through various models. So there's a lot of different aspects to this work, but for today, I'm really actually going to focus on one specific question that we wrestle a lot with that might actually sound quite simple, which is how do we determine how much of a modification is present? And again, that sounds like a very simple question, but what exactly does that mean? Because we can think about that in a few different ways. And so one way we can think about it is what I would sort of refer to as sort of in bulk, and Sara just referred to this in her talk, where essentially if you think about an RNA as just a series of A's, G's, C's, and U's, you can think about how many different modifications you have sort of within that sequence. So for just a hypothetical here, I have an M6A and an M5C. And so then what you can do is think about essentially the ratio of the modified to the unmodified base. And so in papers, you'll often see this expressed as either a ratio of M5C to C, for instance, or M6A to A. Or sometimes people express it as a percentage. And essentially that just is an indication of the sort of overall abundance in the mix of RNA that you're looking at. And the way that we would typically measure this is through mass spectrometry measurements. And I won't go into sort of the nitty-gritty technical details here, but this is essentially an experiment where we would take an RNA pool of interest and we would then digest it first into individual nucleotides, which we then dephosphorylate into individual nucleosides so that we have a mix of individual modified and unmodified nucleosides. And we then inject that onto a liquid chromatography system coupled to a mass spectrometer to perform tandem mass spectrometry. And what we're doing here is essentially just comparing multiple different chemical properties of each of these modifications, including sort of the retention time of each modification on a reverse phase column, as well as different fragmentation patterns. And this allows us to compare to different standards to then calculate the percentage or the ratio of the modified to the unmodified base. And so the data will end up looking something a little bit like this oftentimes. So in this case, let's say we're hypothetically interested in the levels of M6A in different conditions in a cell line. You would essentially compare your different conditions and plot sort of the M6A to A ratio or percentage, and hopefully, you would see some effect that you are interested in. And so there's sort of a few different versions of this that people will use, but sometimes you'll see it expressed this way in a publication. But at the level of a specific transcript, that gets a little bit more complicated, because if you think about sort of the pool of RNA in a cell, we can use our same hypothetical sequence. But if this was, for instance, an mRNA, you would see a mix of different molecules, even within that one transcript of interest, where you might have one modification that appears at, let's say, 3 transcripts out of every 10, and one modification that appears in 5 transcripts out of every 10. And so this is actually what we mean when we refer to stoichiometry. And so we oftentimes refer to this as a percentage. So in this particular case, I would say mod 1 or M6A is at 30% stoichiometry in the sample, and mod 2 or M5C is at 50%. And so measuring the stoichiometry of each of these is actually still quite challenging. And Sara just introduced sort of her fantastic work on direct RNA sequencing. And I think this would really be a dream experiment where, with further development, we could essentially just take our RNA samples, put them, on the nanopore, and then just find, direct unbiased sequencing of RNA modifications in a single experiment. And this, at least for me, somebody who thinks about this a lot, would be a dream to be able to do this. But unfortunately, we can't quite do this to the extent where we can just simply call any different modification we're interested in just without sort of any sort of further development. And so this is something that obviously many labs are actively working on, and we're hoping that we see some great advances in this, and I'm sure we will. But really, in an ideal experiment, what we would want to do is measure modification stoichiometry. We'd be able to measure multiple modifications in a single molecule, and we would be able to unambiguously determine exactly which chemical modification is there at any given time. And unfortunately, we can't quite do that yet with direct RNA sequencing. But just to give you a sense of sort of where some of the other current methods are at, I'm going to use a comparison with a method that a lot of people use to map different modifications, which is antibody-based sequencing. And so antibody-based sequencing is essentially where we just take specific antibodies to a modification of interest. So I think Sara referred to the anti-M6A antibody that's become quite popular. And you pull down your modified RNA and you use high-throughput sequencing to just figure out all of the modified RNAs in your sample. And when you do that, you'll get data that looks a little bit like this. So if you zoom in on a section of your favorite gene, you'll see sequencing reads that pile up, over specific sections in your immunoprecipitated sample, indicating that somewhere under this peak is an M6A for in this hypothetical. However, this is a very easy to implement method. And that's actually why I think it has been so powerful, is many different labs have been able to use this. And it is powerful in that it can enrich low abundance RNAs. But the drawbacks include that these locations are approximate. And so these peaks can be quite wide, anywhere from 100, 200, or even bigger nucleotides wide. This is also not quantitative. So an IP experiment will, you can sort of approximate whether there is more or less in different samples, but it's not going to be able to tell you whether it's 10%, 30%, 50% modified. And then on top of that, antibodies also have some specificity issues where we see crosstalk with closely related modifications. And furthermore, because we've sort of averaged everything out, we can't distinguish between sort of a few different types of modification patterns. So for instance, in this case, let's say we have 2 peaks that we're interested in. We can't tell in an antibody-based experiment like this whether this means that there are some subset of transcripts that have 2 modifications and some that have none, or if there's in fact 2 different sites that can be modified, they're a little bit redundant, but every transcript has one, they're just in slightly different places. And we don't really know whether there are functional consequences of these different combinations because we can't distinguish them. And so you might say, Siggy, well, that seems awfully detailed, does that really matter? And I'm going to actually argue that it does, and that we should care about modification stoichiometry. And hopefully I can convince you of the same in the next few slides. So if we think about this in the context of gene expression, we've talked a fair amount in the last couple of talks about how modifications on RNA can impact RNA translation and structure and other functions. We have to remember that within this central sort of RNA pool in the central dogma, there are many, many different types of RNAs, as has been already alluded to today. And different types of RNAs have very different sort of overall levels and types of chemical modifications. And so, for instance, if we look at tRNA and ribosomal RNA, these are very heavily modified molecules that typically have very high stoichiometry modifications. And so if we use tRNA as an example, we heard about this a little bit in the first talk. Every one of these sites represents a possible modification site. And in fact, in tRNAs are some of the most heavily modified RNA molecules. And interestingly, in many cases, the modification is present at close to 100% stoichiometry because that modification is in fact important for tRNA folding and structure. So it can sort of fold in on itself into a functional form. And similarly for ribosomal RNA, we often see the same thing. But this is not necessarily the case with mRNA. So when mRNA is transcribed from DNA, we often see that we see that there are many different steps that have to happen before the mRNA can actually make its way through all of these steps out into the cytoplasm to be translated. And it turns out that we know now that it is we either know or strongly suspect that there are modifications involved in every one of these different steps. And so once you start to think about what it means if, for instance, an M6A modification is required for export or something like that, you might start to imagine that understanding the sort of stoichiometry that we were talking about earlier could actually be quite important. And so what do I mean by that? So what I mean is that if we think about an mRNA being transcribed and perhaps that modification is installed co-transcriptionally, which we know in many cases is true, it's possible that that modification is critical for the export of that transcript into the cytoplasm. And perhaps when it's not modified, it gets retained in the nucleus. So that means that this modification is essential for export and therefore essential for the mRNA to become available to translation machinery to actually be translated into protein. And then furthermore, we know that there are many modifications that can impact mRNA structure and decay. And so it's possible that additional modifications are installed either in the nucleus or later that will regulate the decay of that mRNA. And so when you look at it this way, you can imagine that perhaps under different conditions, you would start to see higher or lower levels of a modification that would regulate an mRNA's transit from its transcription machinery into the cytoplasm so that it can be translated. And the only way we can really delve into that in a very detailed way is to really understand, what pools of transcripts are modified to what extent, what stoichiometry, and how that correlates with their location and status in the cell. And so just getting back to sort of our antibody experiment, in an antibody-based IP experiment, these are incredibly powerful for understanding sort of what transcripts are modified under some, specific condition you might be interested in, but they won't be able to give you this level of information that could tell you perhaps whether this is a highly redundant modification that can be installed on many different parts of the mRNA, or perhaps there's a subset of RNA that's being retained in the nucleus or being exported in the cytoplasm more effectively. Okay, so I've talked a lot about stoichiometry and why I particularly think it's interesting. So hopefully I've convinced you that this is an interesting problem to study. And so Sara just told you a lot about how the power of direct RNA sequencing and the sort of strides that her lab and others have made in sort of improving those methods so that we can start to get at stoichiometry. But in the meantime, we in our lab also use some alternative approaches to try to get at this question and potentially to provide complementary approaches to direct RNA sequencing in the future. And so I'm just going to highlight a couple of these in the last few minutes here, these top two, but I'll just highlight that there are sort of many other approaches out there that one could consider. So one way we can consider trying to measure stoichiometry is using reverse transcriptases or enzymes that are converting our RNA back into cDNA, but that are special in that they respond to different modifications by introducing mutations. So what that means is that when this reverse transcriptase encounters a modification in the RNA, it will in fact not quite know what to put there, so it will introduce a mutation that is mismatched and not complementary to that RNA sequence. And so you can imagine that if you then sort of continue to reverse transcribe all of your RNA, you can start to pick up possible modification sites by these increased areas with higher rates of mutation. And I'll just point out that there are many different reverse transcriptases out there, and they all have different behavior depending on the modification, the conditions you're using, and things like that. And so depending on your application, you could sort of tinker with it to try to identify one that's appropriate for your modification of interest. If you then couple that to high throughput sequencing, you could essentially get a really sort of large, sort of broad view of where all of the mutation signatures are in your RNA sample, essentially using the mutation rate as sort of a proxy for the modification stoichiometry. But I will highlight, as Sara did in her talk, that controls are incredibly important in these methods, as with any experiment. And so, for instance, in an experiment like this, you must account for many other sources of mutations, including mutations in the DNA itself, sequencing artifacts or RT artifacts, as well as modification specificity. So you need to layer in controls that allow you to pinpoint sort of exactly which modifications you're looking at. But this is sort of a broad class of methods, and there's a few sort of examples of this out there now that allow you to sort of get at stoichiometry in a way that's sort of somewhat similar to direct RNA sequencing, but using a reverse transcriptase and looking at the cDNA product. All right, so just in the last kind of minute and a half, 2 minutes here, I'm going to talk about another way that we are trying to approach this question in the lab, which is measuring stoichiometry via targeted mass spectrometry. And so the idea here is that when you have sort of a big pool of RNA and we digest it for mass spec, we get a big sort of average abundance of a modification of interest. But what we've been trying to do is actually isolate specific fragments of, in this case, pretty abundant RNAs, essentially by annealing a DNA oligo. And we then digest away all of the sort of flanking single-stranded RNA and isolate just that fragment to then analyze that fragment by mass spectrometry, the way we would sort of a bigger RNA pool. And the idea here is that since you know the sequence of this fragment, you could actually do some back of the envelope calculations and guesstimate what your sort of expected modification abundance would be if it was at 100% or 50%, et cetera. And so we've actually done this for a highly abundant RNA, 18S ribosomal RNA, and some work that was published last year. And this is actually a site that was alluded to in the first talk as well. So there's two dimethylated modification sites near the end of the 18S ribosomal RNA, as well as a neighboring M6A site. And so our probe sort of flanked this sequence region right here. And when we performed mass spec on the fragment that we got from this, we found dimethylated 6A at a range of, I guess you could say, about 12 to maybe 20%. And in fact, somewhat surprisingly to me at the time, the expected ratio you might expect based on just the sequence is actually about 18%. So we were quite pleased with that. However, when we tried the neighboring M6A sequence, we weren't quite as good at it. And so clearly there's additional further development to do here. But what was quite cool was that when we knocked out the methyltransferase that installs this M6A right here, these knockout cell lines, which are all shown in red, you can clearly see that M6A disappears. And so the sort of more targeted mass spectrometry approach could provide a really interesting tool moving forward, but it will require a lot of additional calibration and development so that we can do it with RNAs that are much less abundant than 18S ribosomal RNA. All right, so I'm out of time. So I will just leave you with the answer to this question. What do we gain by understanding modification stoichiometry? Potentially quite a bit, at least I think so, particularly when we consider it in the context of the whole cell. And so hopefully you'll come up with some great new ideas and projects and approaches to tackle this question moving forward. And with that, I'd just like to thank my amazing lab, my funding sources, and all of my collaborators. And of course, I'd be happy to take any questions along with the rest of the panelists.
Wonderful. Thank you, Siggy, as well as to Sara and Ya-Ming. The first question we have in the Q&A is really related to, I think, controls and how they're synthesized. And so maybe, I don't know if Ya-Ming or Sara, you want to take that. So really, we don't have the ability to biologically synthesize modified RNAs, right? Really. So we can make unmodified RNA much easier than we can make modified RNA, except with chemical synthesis, and that provides a real constraint on this. I don't know if one of you want to elaborate on that a little bit.
Ya-Ming, why don't you go for it?
Yeah, so my lab has been working on this problem. It's a very important problem. We need standards of modified nucleotides in order to quantify during direct sequencing of RNA using nanopore. So as Mark mentioned, modifications are difficult to generate chemically. So especially for long RNAs. So what we have done is that we will assemble 3 pieces of RNA that together will generate a KB long RNA that will be suitable for nanopore sequencing. But during this assembly process, we'll use enzyme in vitro transcription to generate a left arm RNA, a right arm RNA, and in the middle, we will synthesize a short RNA that contains a site-specific modification using solid phase synthesis. So with this approach, we just published the paper. We are actually able to get a decent yield that affords subsequent biochemistry analysis.
Wonderful.
I just want to piggyback on that just really quick. Everyone is always surprised when they hear that RNA synthesis is not as good as DNA synthesis, because we can synthesize, thousands of nucleotides of DNA, no problem. And it doesn't cost very much money. But RNA, when you go over 25 nucleotides, just for a normal RNA, your yield and your quality goes way downhill. And then when you throw in a modification, I'm just going to throw out a number there, adding a single modification in the center of a 20-mer, upwards of $400 for that one little guy that you're sticking in there. So the reagents are really expensive and the synthesis itself is surprisingly horrible. So not a commercial option.
Although it is one of the topics that NHGRI is funding in terms of a nucleic acid synthesis program. So stay tuned, folks. Hopefully there'll be some new advances in this area. Sara, we had another question about the RNA imaging in your group. Just can you clarify? I think I understood that what you're looking at is the location of the RNA that you pre-identified as being modified, but you're not specifically probing the modified base in those transcripts. Is that right?
We're developing methods to probe the specific base. What I showed you was just a general, what we're doing is we can make this semi-quantitative call that some sites are almost at 100%. So there's some of these unique sites that are almost 100% of the targets are going to have the mod on them. So in those cases, the RNA is representative of a modified RNA. So those dots you were seeing are all of the mRNA, we're not looking at the modified site.
Right. Obviously, lots of interesting stuff to come there. We are at the top of the hour. Thank you, everyone who participated. If you have questions that you didn't get answered, feel free to email us at tdcc@jax.org, tdcc@jax.org, and we'll pass them along to the speakers. Please join us again on Friday for single molecule protein sequencing, and next week for the other talks as well. Enjoy the rest of the day, folks. Thanks for joining us.